our exam 3 review. 4 SIs are going to be doing it today, and we're recording right now. So we'll put that out probably by tomorrow. If you guys have any questions while we do this, just put them in the chat, and whoever's not presenting, we can answer those for you. I think Keyshawn, you can go ahead and get started.
All right, sounds good. I'll start sharing my screen. All right. Can you guys see this? Yeah. Okay. All right.
Yes.
So let's get started. Okay. So for lecture 27, there's not too much on lecture 27 because it's kind of just like an overview of things that we'll kind of go over. When I took the class, this wasn't even on my exam. So I would think of this lecture as like low yield in terms of like overall content. But it does go briefly about like the concepts that you need to know like when studying these pathways. So intro to metabolism. So metabolism, as we know, is going to be like the overall process, right? It's going to be the sum of basically the processes in living systems that living systems get and use free energy to carry out these functions. And so one thing that I would recommend like when studying for this exam, since there's a bunch of pathways and they're all kind of like interconnected, is draw something like this. It doesn't necessarily have to be like a diagram exactly like this, but something like this just helps in understanding like how the different pathways are connected, because sometimes they might ask you questions connecting like glycolysis and gluconeogenesis. So it's just important to know how they're related, or, which molecules obviously enter the citric acid cycle and things like that. And this is also just another chart that I told my students, but something that I feel like you guys should also look at is the location. So the location of these processes are frequently tested. They love asking questions like, and just differentiating between where processes occur. So, like glycolysis obviously occurs in all cells and all parts and all organs. But like something like gluconeogenesis is typically going to be in like the liver. And so this is just a popular table that when you guys are studying, kind of refer back to. And so in terms of chemical reactions, I just want to quickly go over, there's obviously reactions that form and break carbon-carbon bonds. You're going to also see like a bunch of rearrangements, and you'll see this in glycolysis, which I'll be covering next. But you have like the isomerases essentially. You have group transfer, right? So obviously if there's a molecule with high group transfer potential, you're going to be transferring that phosphoryl group in order to make that ATP essentially. And then you also have oxidation reduction reactions as well. So in terms of like the key sources, is kind of cut off, but like the key sources of energy. So we'll typically see ATP hydrolysis as a main kind of factor for driving like the reactions, right? And so you can see it imparts basically a large amount of energy, the free energy that's able to be used to drive these reactions. And so I'm pretty sure you guys are not responsible for knowing like the formulas and stuff like that. Like you guys had to find like, for calculations for concentration gradients and stuff like that. But just know that obviously concentration gradients are a free energy of transport. And also one of the major things that you also see in the electron transport chain is that these electrons that are going to be in the form of those reduced carriers that carry those electrons are going to be used as valuable sources of energy. So the roles of ATP, obviously we're going to consume ATP. 2 ATPs, for example, are consumed in the preparatory phase of glycolysis. And after obviously that second investment of ATP, you're in that, that's kind of like the committed step, you're committed towards glycolysis. But essentially you also have interconversions of these nucleotide diphosphates and triphosphates that is able to occur. And also you just have ATP hydrolysis in general. And so for the formation of ATP, one thing that I want to emphasize on that you guys will see is substrate level phosphorylation. You'll see that in like glycolysis, for example. And what that basically means is that an ATP is basically transferred. The ATP supplies like a phosphoryl group. And so what happens is you have a transfer of this phosphoryl group right here from a substrate. So this is, for example, your substrate, and you have a phosphoryl group here, and it's going to be transferred to ADP to make ATP. And so it's phosphorylation due to a substrate. That's why it's called substrate level phosphorylation. And then you have an adenylate kinase reaction. This isn't really important, but just know that you can kind of convert from AMP to ADP, and then this ADP can essentially be used in a reaction like this, in that like substrate phosphorylation, level phosphorylation. That's why we kind of undergo this conversion. And so in terms of the roles of ATP as well, we also have seen an oxidative phosphorylation. So obviously this is in the mitochondria of animals, and then you have photophosphorylation, which is in like the chloroplast of plants. That's not really important, but the main kind of thing, and I don't want to get into the details of this because we also have like a lecture on ETC. But essentially, like your electrons are going to be flowing from these carriers, right? And electrons obviously have a certain amount of energy, right? So as they're flowing, you have energy available, and that energy is going to be used to pump protons from that matrix to the intermembrane space. And then obviously, like once you have a high concentration of protons in your intermembrane space, you can go from high to low. And ATP synthesis uses that proton gradient in order to generate ATP. So phosphocreatine is important because it kind of serves as a high energy reservoir. So in this like picture right here, you can see like if you have a low energy status, right, you obviously want to combine phosphocreatine and ADP to make ATP, and this is using creatine kinase. And typically, like for brain and muscle cells, they require like high bursts of energy under anaerobic conditions. So those that require like high bursts are typically going to use this kind of reaction essentially. And you can see like the amount of ATP that's being like made. So initially, like when you have ATP, it quickly gets depleted. You can see that through like the graph here, right? And phosphocreatine is good in terms of like supplying that ATP and replenishing that, but also you have like the glycolytic pathway as well. And then you also have like oxidation of fuel molecules such as like, beta oxidation and stuff, which as you guys have studied, does create a bunch of ATP. So lastly, for the fates of glucose, this is just something I want to kind of show you guys. So obviously can literally go anywhere. Glucose can be oxidized in glycolysis, and it can be oxidized to pyruvate. You can also have oxidation via the pentose phosphate pathway, where you're going from the 6-carbon glucose to 5-carbon sugar. You also have glucose as a form of storage. So if you guys remember when studying like Purich's lectures on all the sugars and stuff like that, we had to study like glycogen, starch, and all that stuff. So it can be used in terms of storage, and also it can be used in terms of making like structural polymers as well. So obviously there's just a bunch of different fates and different directions in which glucose can be headed. So for glycolysis, glycolysis is obviously a lot, and it's obviously frequently tested. So I'll get right into it. And so for glycolysis, it's going to be the glucose oxidation, right? That's basically what we're doing is we're oxidizing glucose, and it's going to be carried out in the cytosol of all cells. And so it actually doesn't require oxygen, unlike the citric acid cycle and your electron transport chain. And so it's important to kind of differentiate like these different kind of cells and organs in terms of like the glycolytic pathway and if they rely on it or not. So obviously like red blood cells, they're going to rely entirely on glycolysis because they don't have a mitochondria. And the reason why we undergo glycolysis in the first place is to, make energy. And so we also have skeletal muscle, which can undergo aerobic and anaerobic. Your adipose. So this is actually a question that I got on my test that I remember. And it's an example of how things can be connected and how they can ask you stuff. So what they asked was basically like which molecule can be made into like a glycolytic intermediate. You basically just had to know that like glycerol can be made into like the glycerol of tag can be converted into dihydroxyacetone phosphate. And so it kind of shows like how you can have a connection between different processes. And so it's important when you're studying to kind of link that together because you could get a question just like that. In terms of liver, so liver, obviously you have fatty acid synthesis. And then for the brain, it's also going to rely on glucose or glycolysis a lot, just like those red blood cells. So these two are going to heavily rely on it, except this is going to be in aerobic conditions. And the reason why is because obviously you have beta oxidation to make energy, but the thing is the brain can't use those like long chain fatty acids in order to get that energy. And so that's why it relies on glycolysis heavily. So just an overview of glycolysis, you're going to have a 6-carbon glucose, right? This is your 6-carbon. You're going to be converting into 3 carbons. You'll see like the glyceraldehyde, 3 phosphates. And then essentially those are going to be then converted into pyruvate. And so when you're looking at like the entire kind of net, sort of like you should look at like what the net products are. You're going to have like your 2 pyruvates coming from that glucose. You're going to have a net ATP of 2, and you're also making NADHs. And these NADHs are important because they'll eventually be used and can be converted into energy. They typically hold 2.5 ATPs. That's how much they're like worth. And so that's important to note that they're worth 2.5 ATPs, and then like your FADH2s are 1.5, and like you'll see that later on. So in terms of glycolysis, obviously just know the structures, enzymes, and stuff like that. But you also have to know chemical logic. What that means is you have to know like why we do certain steps. And it's kind of like whenever you're going through the pathway, you should kind of ask yourself like, why are we doing this step in the first place? And I'll kind of walk you through that for each of the steps. But essentially, a mnemonic that worked for me for the enzymes was high fat, don't kill me, please. So if you guys want to just memorize the enzymes in terms of that, you could use that. And then in general, what you'll see is a kinase will typically phosphorylate, and it'll be like involved in that like substrate level phosphorylation, for example. And then your dehydrogenases are typically going to be they're going to oxidize your substrate, and they're going to be involved in your, like whenever you see a dehydrogenase, it's typically going to be involved with like an NAD plus or like a reduced electron carrier. And isomerases are just going to be interconversions that you're going to have between isomers. And then obviously your net is going to be your 2 ATP, 2 NADH, and also what I didn't include here, but 2 pyruvates as well. So for the first step, you have something called a preparatory phase, right? So you have a preparatory phase and a payoff phase. So for the preparatory phase, that's going to be your first step, and you're basically going to phosphorylate glucose. And so glucose is going to be phosphorylated into glucose 6-phosphate through hexokinase. So you guys, I'm pretty sure you guys already learned this reaction from, I think, exam 1 about hexokinase. And so it should be a little familiar, but basically the reason why we do this is to capture glucose within the cell. And so what that means is essentially once it's phosphorylated, like glucose 6-phosphate can't leave. There's no transporter for it to get kicked out. And so this kind of captures it and helps it kind of go into this like phase of glycolysis. But the thing is, the first step is not going to be the committed step in this case. And the reason why is because glucose 6-phosphate can literally go to any other, like it can go to like other pathways, such as like the pentose phosphate pathway. So technically it's not like loyal to glycolysis. It could go to like the pentose phosphate pathway because that is like the starting molecule for that, for example. And so for the second step, it's going to be a conversion, right? And so you're going to use an isomerase. So typically when you see conversions such as glucose 6 to fructose 6, think of those isomerases. That's just a good way to see it. And so what you have is you're essentially converting like the location of this like carbonyl from the first carbon, and you're going to move it down to the second carbon. You're basically just swapping these two. And so the reason why we do this is because we want to phosphorylate this N carbon. But if you have a carbonyl here, like you can't phosphorylate it. And so essentially you want to move it because you have an OH here, and this OH can then be phosphorylated, right? It can be attacked. And the reason why we want to phosphorylate is because if you have a phosphoryl group here and another one here, you can literally cleave the bond, right? You can cleave it, like you can split the 6 carbon into 3. And so that's basically what we're doing. And so for phosphorylation, obviously you're going to phosphorylate this, and so you're going to have that phosphoryl group here. And so you're going to go from fructose 6 to 1,6. And just a good way, if you guys want to see like the names, if you're asked like this molecule, right, and you're asked like what the name of this molecule is, you can see like it has 2 phosphoryl groups, and you can number the carbons like it's going to be on your 1 and 6. That's why it's fructose 1,6. And fructoses are going to be that 5-membered ring. And so that's just a good way to look at it in terms of if you're asked about like a structure, for example. And this is actually going to be your rate limiting step, and this is going to be through PFK1. The reason why this is the rate limiting step or the committed step is because now you've invested a second ATP, right? So in that first reaction with the hexokinase, you're using 1 ATP, and now you're using your second ATP. And so once you kind of used up that energy, you're kind of committed to this process. And you can see like the delta G is going to be like really negative. And so this is kind of like a irreversible step essentially. And so the reason why we do this is because now we have a symmetric molecule. We have a phosphoryl on the first one and the sixth one. So now we can cut it. And so now we have cleavage, right? So that's the next step. We're cutting the molecule through an aldolase. And so you're going from fructose 1,6 to 2 molecules, right? So obviously 2 3-carbon molecules. You have dihydroxyacetone phosphate, or DHAP, and G3P, or glyceraldehyde 3-phosphate. And so the cleavage requires a carbonyl at the second carbon and a hydroxyl at the 4th carbon. And so that's basically like why we, this is like why we basically cleave it, is like in order to break it into these two. And what happens is actually The delta G here is super positive. So you might encounter on your exam, like they might ask you like why a certain delta G, why we even go through the reaction if the delta G is positive. Like they don't teach you this in class, right? But you have to like in lecture, he kind of told you that you make a low amount of your you're making a low amount of G3P, and actually G3P is the molecule you want to continue glycolysis. So since you're making a low amount of this, it's actually going to shift like through like the equilibrium stuff, Le Chatelier's principle stuff. Like you can see that the equilibrium is going to shift towards products. You want to make more of this. And so you're essentially driving this. And so like the overall delta G is going to be negative essentially. So that's like the reason why. But that's just an example. Like you don't have to know the reason why all the delta G's values are like negative or positive and stuff like that. I would kind of focus on probably this one since it's like really big. But that's like the reason why. And typically you'll see a bunch of like hypothetical scenarios, which is kind of annoying. But if you understand kind of like the basic process, you can reason through like what would actually happen if, I don't know, a certain enzyme didn't function properly. So he likes asking hypothetical questions for this exam. But anyways, you have 2 3-carbon molecules, right? And so that's what we have. And so as I said, we have a low amount of G3P. So we actually want to convert your dihydroxyacetone phosphate that you have to G3P, or your glyceraldehyde 3-phosphate. And this is going to be through that like triose phosphatase. And so this is actually going to be your last step for your preparatory phase. And the reason why is because now we have the molecules that we want in order to continue glycolysis. And so your 6th step is essentially going to be your oxidation, right? And so you have your oxidation of your G3P to 1,3-bisphosphoglycerate. And so that's going to be through a dehydrogenase. And so typically dehydrogenases, we associate them with NADH and NAD+, right? And so in this reaction, what's happening is your glyceraldehyde 3-phosphate combined with an inorganic phosphate, like a free phosphate, is going to be oxidized to your 1,3-bisphosphoglycerate. And so as this gets oxidized, your NAD+ is going to be reduced to NADH. And so the reason why we do this is because now you have a phosphoryl on here, right? So due to that free phosphate, that free inorganic phosphate. And so this molecule right here has high phosphoryl group transfer potential, meaning like you can use this in order to do substrate level phosphorylation. Like you can donate this to an ADP in order to make ATP, if that makes sense. And so we'll see in the next step, essentially, you have now your first substrate level phosphorylation. So you can see like, as you're going through each of them, it like makes sense why we're doing this. And you should go through when you review this, like why we do each step. And so you have 1,3-bisphosphoglycerate, right? And you have ADP, right? So that ADP is essentially going to be turned into ATP. And so you're going to have this donation of this phosphoryl group from your substrate to ADP. And so you're making ATP. And since you're donating you're donating that phosphoryl from that first carbon, you're cutting that first carbon off, and now you have 3-phosphoglycerate. So you can see like within the name also it makes sense like for the molecule. So it's not too bad if you think about it in this sense. And like this is obviously going to be catalyzed by a kinase. It's going to be phosphoglycerate kinase because you're phosphorylating, right? Kinases phosphorylate. And so at this point in the step, you have 2 ATPs made and 2 that were used up. So 2 were used up in that preparatory phase, and now 2 are made essentially. And so this is an energy coupling kind of reaction. And so like the formation of that bisphosphoglycerate, the delta G for this was actually positive, but through ATP hydrolysis, it makes it negative. And so you can see like the ATP, like it's super like negative. So that drives it into a favorable, like it's the whole coupling stuff essentially. And so after that, you have a conversion. And so we've never really seen mutases before. This is kind of like the weird thing where like we've never really kind of looked at it before. But anyways, mutases are going to transfer a functional group. And the reason why we do this is to release water essentially. And so you have a 3-phosphoglycerate, right? So you went from 1,3 to 3. So now you're essentially just breaking it down, right? So you're going from 3-phosphoglycerate now to 2-phosphoglycerate. And the reason why we kind of convert and transfer this is to remove water. So you have your phosphoryl on that 3rd carbon, you're going to move it to your 2nd one right here. And so when you do that, you have an H, a hydrogen right here, and you have an OH right here. So these 2 are going to combine to remove water essentially. And so in the next step is the dehydration step. So you have these 2, and you're gonna remove water. This is gonna be through an enolase. So enolases typically make double bonds. That's just kind of like a general rule of thumb. And you'll see with the removal of water, you make that double bond right here. And so you go from 2-phosphoglycerate to phosphoenol pyruvate. And the thing is with this now, when you transfer your phosphoryl from your third carbon right here, that was initially here to here, now you have a molecule with high group transfer potential again. So what happens is anytime you make a molecule with high phosphoryl group transfer potential, the next step is literally going to be substrate level phosphorylation again. And so we look at that last step and we see that again, it's going to be your second substrate level phosphorylation. And so you have your phosphoenolpyruvate with your ADP. And essentially that ADP is going to be turned into ATP. This gets donated to ADP to make ATP. And then this through pyruvate kinase, obviously this is the kinase phosphorylating, and then you make your pyruvate essentially, and that's going to be your primary product. So you're going to be making a total of two essentially from one glucose molecule. And so in terms of the fates now, so once you have pyruvate, there's different things you can do. I wouldn't focus on the fermentation to ethanol really. You won't really see any questions on that. But the two main ones that you guys should know is that pyruvate will essentially go through like that PDH complex to make acetyl-CoA, and then that goes to the citric acid cycle. And like per acetyl-CoA, I believe it's about 10 ATPs, right? So each cycle is you're making 10 ATPs. And then you have your pyruvate, and it's going to go and convert to lactate. And so that's an important reaction that I want to go over. And that's going to be in anaerobic conditions. So this is for sure a reaction that you guys are responsible for, that you'll for sure kind of see on test day. And the reason why, and I'll kind of explain why, because it's really important in kind of undergoing glycolysis again. So you have something called homolactic fermentation, right? And so if you typically do like, say if you do like a HIIT workout, right? Like a high interval intensity workout. And so you essentially have you need like energy essentially, right? So you need you need a lot of energy. These muscles are typically going to switch to anaerobic respiration because it's just quicker to make energy. And so as a result, you're actually making lactate as well as a byproduct of your anaerobic respiration without oxygen. And so lactate is going to be formed in your muscles. It's going to be released into the circulation and reconverted back into glucose in the liver. So essentially what's going on here is you have pyruvate, right? So this is after glycolysis, and through lactate dehydrogenase. So obviously dehydrogenases are going to use those like reduced carriers, or it could be oxidized carriers depending on what's going on. So in here, your NADH is becoming oxidized to NAD+, your pyruvate is going to be reduced to lactate. And so the reason why we do this is now that NAD+ can be cycled back into glycolysis, right? So we use NAD+ in order to convert it to NAD. As I said before, it can be released and converted back to glucose in the liver. Glucose is literally the starting material, right, for glycolysis. And so you can undergo glycolysis again. So that's why this is an important reaction, is to kind of continue glycolysis, that cycle. So this is just a question for you guys. You guys can just read the question and then put the answer in the chat box, and I'll kind of go over it. Yeah, good job. So it looks like a lot of people are saying B, and so that's actually correct. And so I'll kind of go over it. But essentially, this is just something that you could see like on your exam. It's kind of annoying because they do require a lot of reading. Kane did mention that like the questions are going to be probably the longest questions you're going to see are going to be on this exam. But if you kind of look at just the last line for most of them, like you can see like which of the following conditions in the body would directly stimulate PFK1 to ultimately stimulate the glycolysis pathway. So obviously what kind of activates PFK1? And so PFK1 is going to be used in glycolysis. And so the thing, if you think about it in general, glycolysis is making energy, right? So low energy molecules are going to be what kind of stimulates it, right? Because if you already have high energy molecules, like there's no point in making more energy. You don't need it. And so if you look at like the answer choices, you can see that if it's high levels of ADP is a low energy molecule, right? So if you have high levels of ADP and low levels of citrate is actually going to be one of those high energy molecules. So if you have low levels of that and high levels of also another low energy molecule, this is going to be your answer, because everything else is going to be like high levels of energy. Like obviously this is going to inhibit the pathway essentially, for example. And so that's why B is the correct answer. So next, this is kind of just a meme for you guys, but next I'll be covering gluconeogenesis. Okay, so for gluconeogenesis, think about basically we're making, genesis obviously means to make, and so gluco kind of precedes that term, and so we're making essentially glucose. And so in terms of gluconeogenesis, it's going to be the reverse of glycolysis, but it's going to have 3 bypass reactions that you guys got to know. They love asking questions about bypass reactions, and it's just easy points if you guys know which ones are regulated and have like a good way of kind of understanding which ones those are. And so gluconeogenesis, if you guys refer back to that lecture 27 table that I went over, you can see that it occurs in the liver. And so the liver actually is going to make glucose typically when you're like sleeping, or if you're long-term fasting, is because the levels are low. So obviously if your levels are low, like it'll release like glucagon, and then that'll kind of stimulate the whole pathway. And so the 2 main places in your body that kind of need glucose are going to be your brain and your red blood cells. Your brain typically favors glucose as a fuel source. And so in terms of like the precursors for gluconeogenesis, you're going to have adipose tissue that breaks down into to TAG, and TAG will be broken down into obviously like those 3 fatty acids and your glycerol, but that glycerol can be converted into dihydroxyacetone phosphate, and that can enter like the glycolytic pathway, but also it's important in terms of gluconeogenesis, because obviously it's just the reverse, right? And so we also have muscles. So muscle cells you can have like the alanine, it can be transaminated to pyruvate. So if you like look at this picture right here, you can see like lactate can go into the liver. So this is like that whole after homolactic fermentation, right? You have lactate, your lactate will go to your liver and it can be converted to glucose. And so that glucose then helps in terms of, you can store it in terms of as glycogen essentially. And so here, like you can see that ATP is being used in order to make glucose. And then here, like alanine, you can have pyruvate convert to alanine. It'll go into the liver and it'll convert to pyruvate. And then that obviously you can start gluconeogenesis with that pyruvate, because pyruvate is that first molecule essentially in the gluconeogenesis. And so that's basically the precursors. Those are important. And so in terms of the bypass reactions, you're going to have pyruvate kinase, your phosphofructo or PFK1 essentially, and your hexokinase. So just a good way to see which ones are the bypass reactions, you're going to see that it's going to be your first step in glycolysis, essentially, your last step in glycolysis, and it's going to be that committed step. So first, last, and committed. Those are the 3 that are going to be replaced essentially with different enzymes. And so I would really know like which ones are going to be the bypass reactions. So for the first bypass reaction, it's going to take place in the mitochondria. And so I kind of put a question here, but like for pyruvate carboxylase, right? So what happens is you have pyruvate. This is like the end product of glycolysis, but we're now going in the reverse direction. So you have pyruvate, and you're going to use bicarbonate in order to break it down into oxaloacetate. And that's going to be through pyruvate carboxylase. But for some reason, they like asking questions on what pyruvate carboxylase specifically requires. So it kind of requires a biotin cofactor, and it also is going to be activated by your acetyl-CoA. And so those are 2 really important facts about pyruvate carboxylase that you guys have to know. And so the next step you're going to have is oxaloacetate, and you're going to be converting it to phosphoenolpyruvate, and that's going to be through PepsiK. And this one's kind of weird because it requires GTP, right? So typically we'd see like ATP or stuff like that, but it's going to be using that guanosine triphosphate in the sense. And so the reason why we make this is obviously you're making like a high group transfer potential like molecule, phosphoenolpyruvate. And so your second and third bypass reaction, so instead of PFK1, we're going to be using something called FPBase1. And so you're going from fructose 1,6 to fructose 6, right? So this is a phosphatase. So you're removing that phosphoryl group from your first carbon, and now you have just fructose 6 essentially. And you can see like in general, like the delta G's for these are all negative. Like these are all favorable reactions in gluconeogenesis. And then your third bypass is going to be, instead of hexokinase, we're going to have actually a phosphatase, a glucose 6 phosphatase. So we're literally, instead of going from glucose to glucose 6 with hexokinase, we're actually going to be going from glucose 6 to back to glucose because we want to make glucose, right? And that's going to be through that phosphatase. And so the overall pathway, this is just to like look at what's going on overall, but you have essentially your two pyruvates, you have ATP, you also have GTP. So you obviously can see that like through gluconeogenesis, you need energy, right, in order to convert it back into glucose. So you're using these these energetic molecules, and you have glucose as a end product, and then obviously you're using like NADH, it's going to be converted back to NAD plus. So everything's in the reverse essentially. And so in terms of regulation, this is important in terms of like, I guess, hexokinase. Glucokinase is not really tested, but hexokinase is kind of more tested. I'll kind of explain what's going on, but you have basically hexokinase in all your cells, and glucokinase is going to be in your liver only. So hexokinase is going to have a high affinity, low capacity. What that means is essentially, if you look at this graph right here, if it has a high affinity, right when it picks up glucose, it's super strongly like bound to glucose. Even a small concentration of glucose will literally saturate the enzyme, right? You have a saturation like a plateau, essentially. And hexokinase is actually going to be product inhibited. So obviously hexokinase makes glucose 6 phosphate. So this glucose 6 phosphate, if it wants to shut down this hexokinase, it will come back and inhibit it. That's that whole end product inhibition. And then you have glucokinase, and glucokinase is going to have a higher affinity, low affinity, high capacity. And so it has a low affinity, meaning like, all right, glucose is binding to glucose, but it's low affinity, but also it's going to be a lot of like high capacity. Like you can see the concentration reaches so high before it like plateaus. And this is not going to be product inhibited. And so in terms of like, I guess the golden rule, I forgot to edit this slide, but a good way, I guess, to kind of think about activation and like whatever's activated and inhibited is energy states in terms of glycolysis. But high energy states, obviously for glycolysis, we're making energy. So obviously if you have enough energy already, like if you have a bunch of ATP, there's no need to go through glycolysis to make more. And so you're going to inhibit it if you have high energy molecules. And then low energy molecules, of course, are going to stimulate it. You can see through this curve, you might get a curve similar to this, and they might ask you potentially like, I don't know, like what is the curve for A, for example, or like for B. And you have to know like if it's to the left of the C curve is the normal curve. These are going to be the activators. These are going to be the inhibitors. And these are just examples, like the activators are going to be low energy molecules, and then the inhibitors are going to be high energy molecules like your ATP and citrate. For PFK1, sorry. And so, and that's typically also just seen in general with all the glycolytic enzymes. And so like this picture, if you see like for A, it's going to be fructose 2,6 bisphosphate. So one of the weird things about glycolysis is that it's actually activated by fructose 2,6. We don't really cover fructose 2,6 too much, but that's just something you have to know. And it's important because it ties with like the bifunctional enzyme, which I'll talk about a little later. But that's one of the big activators of glycolysis is that fructose 2,6 bisphosphate, aside from like those other low energy molecules such as like AMP. So you can see like the curves, these are the activators, and then these are obviously the inhibitors. But if you look at something like D, right? D, you have an inhibitor and an activator, right? And so typically, if you have a combination of both, it'll still be kind of to the right, but it won't be all the way to the right when you have like ATP or citrate. It'll kind of be like a little less to the right. And so in terms of hormonal regulation, I don't want you guys to think about it in terms of this, because this can obviously get a little confusing. If a kinase is phosphorylated, it's inactivated. That doesn't always work. It can actually be inactivated or activated, so don't follow this. I accidentally included it in there, but and this is how I want you guys to kind of think about regulation, because it's not too bad and it makes sense. And so if you have something called like pyruvate kinase, right, this is that last enzyme in glycolysis. If you have low blood glucose, right, it's going to obviously lead to like glucagon, the stimulation, the release of glucagon. Glucagon is going to bind to its GPCR receptor. And the thing is, you don't have to know like the whole like pathway again about like that kind of stuff. Just know that like obviously you release glucagon, it's going to activate pKa, and kinase is always phosphorylate, right? So pKa is always going to phosphorylate. But if you don't know if it makes it inactive or active, just think about it. If you have low blood glucose, like if you don't have enough glucose, and glycolysis obviously requires glucose, you're not going to want to go through glycolysis So phosphorylating this is just going to inactivate it. That's kind of how I thought about it. So you don't confuse like the whole kinase phosphorylated inactivation or activation, because it's different for each pathway. And then for a high blood glucose, you're going to have insulin again. Insulin binds to its receptor. You have PPI, or protein phosphatase 1. And obviously, a phosphatase always dephosphorylates. And so when you're dephosphorylating, it's going to activate it. And the reason why is because if you have a bunch of glucose, you want to go through glycolysis, right? Glycolysis requires glucose. And so if you have a bunch of glucose, you can just break it down in order to make that energy. And so now in terms of regulation for gluconeogenesis, this is a little annoying because it's just like a little confusing. And so I'll kind of go through it, but you have your PFK1, right? So PFK1, its bypass reaction is going to include your FPBase1. And so the FPBase, if you think about gluconeogenesis, it needs those high energy molecules. So it's going to be activated by high energy molecules such as ATP, GTP. It requires ATP and GTP, as we kind of looked at. And the inhibitors are going to be those low energy molecules. And so you remember I said glycolysis is activated by fructose 2,6-bisphosphate. So actually the levels of this are actually controlled by something called a bifunctional enzyme. The reason why it's called bifunctional is because it has two functions. It has a kinase activity and has a phosphatase activity. And so if you look at like in terms of these two scenarios here, because typically these regulation questions are going to be scenario based. So you'll have something called like low blood sugar, right? If you have low blood sugar, that's going to lead to an increase of glucagon again, then PK. PK is going to phosphorylate. But if you don't know if phosphorylation of the enzyme will lead to like a stimulation of like what, you can think about it. Like if you have low blood sugar, you're gonna want to make glucose. So you're activating gluconeogenesis. And so if you phosphorylate your bifunctional enzyme, you're actually going to be activating that phosphatase. So what a phosphatase does is actually you're going from fructose 2,6 to fructose just 6, right? You're basically popping off like a phosphoryl group. And so if you decrease this, it's going to stimulate gluconeogenesis. But if you increase this, it's going to stimulate glycolysis, which is seen here. And if you run through the scenarios, the reason why I highlighted the beginning and end is it matches up. Like if you draw this kind of pathway out, you can see that it matches up like low blood sugar, gluconeogenesis, high blood sugar, glycolysis. And you can see that once you have insulin, then you have PPI, that's going to dephosphorylate, and that's going to activate your kinase. And so you're going to go from fructose 6 to fructose 2,6. And anytime you have a high level of fructose 2,6, you're going to stimulate glycolysis. So it all makes sense essentially if you run through it and see like in terms of like why it like kind of makes sense and stuff like that. But this is just an example of something you could see. Yeah, there's another question you guys can answer. Just yeah, good job. So it looks like a majority, like a lot of people in the chat are saying B, so that's good. B is the correct answer. And so I'll just go quickly right through it. I think this is like my last slide, but basically when you're asleep, you're gonna have low blood glucose, right? You haven't eaten a meal yet, so of course your levels are gonna be quite low. And so if you have a low level, you're gonna have that glucagon, then you're gonna have your pKa, and you're going to have that pKa phosphorylate your bifunctional enzyme. And if you think about it, low blood glucose stimulates gluconeogenesis, right? So you don't want glycolysis. And so you're actually going to want to decrease the levels of fructose 2,6. And the only thing that decreases fructose 2,6 is a phosphatase. And so that's going to be activating your FPBase too. So if you want to think about it in a reverse direction, you can. Like you can just think about it like that. It kind of just helps in terms of understanding it. And so if you have a low level of this, you're obviously inactivating the enzyme required for glycolysis because you don't want to go through glycolysis. And so you're decreasing the rate, obviously, of glycolysis, and you're increasing the rate of gluconeogenesis. And you can kind of see, like, if you don't know which ones are which, like you can see like, okay, if you're asleep, it's low blood glucose, would you decrease the rate? Like you can easily rule out like answer choice C if you don't even know any of like these, right? So I would definitely use that on the test in terms of just like a strategy. And that's basically it. So I'll pass that over to Danielle.
Thanks, Keyshawn. I'm going to share my screen now, or try to. All right, great. All right, so chugging right along to lecture 30, I'm going to be covering briefly more about glycogen metabolism. Okay, so essentially what we're covering in this lecture is all about the storage and mobilization of glucose. And as we learned in this lecture, glycogen is the storage, the polymeric form of glucose. So yes, it is kept in the muscle cells for energy. It also serves a storage function in the liver. And it's really important to know that this is essentially the storage form of glucose. In essence, that it's a really great way to store glucose in our cells because it will avoid osmotic pressure. And what Dr. Xylee teaches us with regards to this is that say if we had 55,000 glucose residues per one molecule of beta glycogen that we have in a cell, we would have an insane amount of water rushing into the cell due to osmotic pressure, just trying to equilibrate that concentration there. So that's essentially what we learned in terms of like the importance of glycogen being used to store glucose in our cells. In the picture here, you can see those tiny little black dots. Those are little glycogen granules, specifically beta glycogen granules. And like I said before, one glycogen molecule contains an insane amount of glucose residues. The internet will tell you about 20,000 to 60,000 glucose residues, but for this class, we learn about 55,000. Okay, a little bit more about the structure of glycogen. So essentially, this picture right here shows a glycogen particle. There is a protein in the middle, which is essentially like the starting point for these chains of glycogen in which we branch out from. So glycogen contains linear chains in addition to branch points. It's important to know that the linear chains of glycogen are alpha-1,4 glycosidic bonds. Meanwhile, the branch points are alpha-1,6 glycosidic bonds. This is going to become important when we start talking about the making and breaking of glycogen in the future. All right, so moving right along to glycogenolysis, which is essentially the breakdown of glycogen. So when we are breaking down glycogen in our cells, this is completed through the actions of the enzyme glycogen phosphorylase A/B. So essentially glycogen phosphorylase A is the inactive form and the phosphorylase B is the active form, which we'll talk a little more about later when I cover regulation of this process. So essentially, the glycogen phosphorylase B will catalyze the phosphorylasis of the non-reducing ends of the glycogen chains at the alpha-1,4 glycosidic linkage. So essentially, when we are making and breaking glycogen, it's important to know that all of these actions are occurring at the non-reducing end, essentially the end points of the chain from that gigantic sphere as we're kind of working our way in or expanding our way out for the molecule. This glycogen phosphorylase AB essentially phosphorylases these glycogen chains to liberate glucose 1-phosphate, which is important to note that this is different than glucose 6-phosphate. And another important point to note here is that this enzyme works through a phosphorylase mechanism. So this isn't a hydrolysis. Essentially what is going on here is that we are using a phosphate to essentially cleave off one glucose from the chain of glycogen. All right. A little bit more about debranching enzyme, which is another enzyme used in glycogen breakdown. So essentially the enzyme that I just talked about, phosphorylase AB, can only cleave alpha-1,4 glycosidic bonds. So we're going to need a different enzyme to break those branch points, which contain alpha-1,6 glycosidic bonds. The enzyme that does this in our cells is the debranching enzyme. It has essentially two activities on that same enzyme. We have the transferase activity, which is essentially going to transfer the glycogen chain ahead of the branch point to another location on the unbranched part of the chain. And then once we are down to essentially one glucose attached at the branch point, we are going to use that glucosidase activity to cleave the alpha-1,6 branch point bond there, essentially liberating glucose at that point. All right, so another enzyme that we cover in this lecture is the enzyme phosphoglucomutase. This enzyme serves to convert glucose 1-phosphate into glucose 6-phosphate, which can readily enter glycolysis, which glucose 1-phosphate cannot do. So that's essentially the importance of this enzyme, is to convert this molecule into something that we can't use for glycolysis into something that we can. So this diagram below kind of shows the mechanism for how this enzyme works. I believe Dr. Zeile told us not to put too much effort into memorizing what's happening here, just essentially knowing that we have an interaction with a serine group on the enzyme. Also, it is important to know that in the liver, this glucose 6-phosphate is dephosphorylated to glucose by glucose 6-phosphatase. This enzyme name is pretty intuitive. It's named after its reactant, and as we have learned before, phosphatases are a class of enzyme that will remove phosphoryl groups. And this is essentially happening, I believe he mentioned that it's occurring on the luminal side of the endoplasmic reticulum. And this is just important because, as we have learned in the liver, this is where we carry out gluconeogenesis. And so we need that glucose in order to have the building blocks for gluconeogenesis. Okay. A little bit about glycogen synthesis now. So we essentially covered glycogen synthesis in a 3-step manner. So essentially we covered that in the first step of glycogen synthesis, we are forming this activated sugar nucleotide. This is very similar to how we have used the hydrolysis of ATP to facilitate reactions in glycolysis. It is very similar here in that we are combining UTP with glucose 1-phosphate and yielding UDP glucose, which is the molecule shown on the upper right-hand corner here, with the liberation of pyrophosphate to essentially make this step favorable. So once we have that UDP glucose, so this step I forgot to mention is catalyzed by UDP glucose phosphorylase. So once we have our activated UDP glucose, then we are all set to begin elongating the glycogen chain. And the enzyme in which we use to elongate the glycogen chain is our glycogen synthase, or as called in the lecture, glycogen synthase AB. This is a highly regulated enzyme. And essentially what we learn about the mechanism of glycogen synthase is that we are essentially liberating UDP as we are condensing these activated sugar nucleotides together to create this glycogen chain. So it is important to note that glycogen synthase creates alpha-1,4 glycosidic linkages. In the third step, we learned about how we synthesize the branch points for glycogen. So essentially, the enzyme that carries this out is the branching enzyme. I love the enzymes when they reflect what they do. So essentially, this is an amylo 1,4,2,1,6 transglycosylase. This is essentially just telling us that we are taking a alpha-1,4 glycosidic linkage and transforming it into that alpha-1,6 glycosidic linkage. As shown in the picture here, we see that we are taking a completely linear chain of glucose here with only alpha-1,4 linkages, and essentially branching off using alpha-1,6 branch points. Okay, so a little bit about the regulation of all of these enzymes, which I just covered. when talking about the regulation of glycogen breakdown, it's important to talk about glycogen phosphorylase AB because this is the enzyme that will catalyze the breakdown of the linear aspects of the chain, essentially the alpha 1,4 linkages. So for glycogen phosphorylase, as I mentioned before, the phosphorylase A is the active form of the enzyme, and the phosphorylase B is the inactive form of the enzyme. So I'm just going to kind of verbalize what this diagram is trying to explain to us here. So essentially, when we have glucagon in our blood, it can signal to the liver. So essentially, glucagon will activate an enzyme which acts on glycogen phosphorylase AB. So we learned that the enzyme phosphorylase B kinase is going to phosphorylate this enzyme, therefore activating it. And in the muscle cells, this happens by a similar mechanism in that when we have high concentrations of calcium and high concentrations of AMP, essentially signaling that we are respiring. This is a very active cell. It's in need of more glucose through glycogen breakdown. Essentially, this will also activate our phosphorylase B kinase to phosphorylate it and activate it to our phosphorylase A. To the contrary, when we are in conditions of low blood glucose, we are going to be activating our phosphorylase A phosphatase, which is on the left, via a mechanism with PP1. And essentially, when we are removing the phosphate groups from the phosphorylase A enzyme, we are therefore inactivating it, essentially turning it into the phosphorylase B form, which is less active. And in doing that, we are downregulating the process of glycogen breakdown. It is also important to note that there is allosteric regulation of this enzyme. So essentially, when we are in high concentrations of AMP, essentially signaling a low energy state, that is going to allosterically regulate our phosphorylase A enzyme, essentially upregulating our phosphorylase A, which should make sense because when we are in conditions of low blood glucose, we want to stimulate glycogen breakdown in order to liberate more glucose. To the contrary of that, when we are in high concentrations of ATP, this is going to work the opposite way, essentially down-regulating that phosphorylase A enzyme. Okay, so as you've noticed, when we've started talking about the regulation of these enzymes, to a phosphatase. So for the enzyme glycogen synthase, I wanted to give you guys this little mnemonic here, which might help you in being able to distinguish this between other enzymes that we've learned. So a mnemonic that our SIs taught us when I was taking this class was get SAD. I feel like it might be a little bit easy to remember on the exam because maybe, I don't know about you guys, maybe sometimes we're sad when we take biochem exams. But essentially the acronym get SAD is telling us that glycogen synthase is active when it is dephosphorylated. So knowing that knowledge, we are going to have the A form of glycogen synthase when it is dephosphorylated. Oops, I'm sorry, I didn't mean to do that. And what enzyme will dephosphorylate glycogen synthase? That is our PP1 enzyme, which is essentially, as we have learned before, is activated by insulin, which is indicative of high glucose levels in the blood. It is inactivated by glucagon and epinephrine, which reflect essentially a state of energy demand. Glucose 6-phosphate will activate this phosphatase and also glucose. If any of this information is confusing to you, I encourage you to, like Keyshawn said, take a step back from what we're looking at and remember that these processes are very intuitive and that For example, here, PP1 is upregulated by insulin, which is indicating that we have high levels of blood glucose. This glycogen synthase is going to want to be active when we have a lot of glucose in the blood in order to start synthesizing glycogen. So kind of taking a step back and looking at, okay, I have these conditions, what am I going to want to do with these conditions? So essentially, that is a really good way to be looking at the activity of glycogen synthase. Also, when this glycogen synthase is phosphorylated, it is deactivated. So in this diagram here, you can see the CK2 and GSK3. Those enzymes aren't super important to know. I know that Dr. Zeile wants us to know that protein kinase A has a very large activity on glycogen synthase. So essentially, when glycogen synthase A is phosphorylated by pKa, we are essentially inactivating it by phosphorylating the serine residues on the carboxyl terminus of this enzyme. Okay, moving on to lecture 31. So lecture 31, we learned about the regulation of carbohydrate metabolism and a little bit about the pentose phosphate pathway. I am going to walk us through an example of how to kind of tackle these exam questions that Dr. Zeile is going to give you all, where we are given conditions and essentially need to be able to logically reason out what enzymes are going to be activated, what concentrations will certain signalers be at, and essentially what is the end result of these biological conditions. So we're going to move ahead. Okay, so essentially I'm going to first cover regulation of fructose 2,6-bisphosphate. So the conditions that we're given is we haven't eaten in a while, so therefore our blood glucose levels are low. So knowing this information, do we want to start gluconeogenesis or glycolysis? So because we have low levels of blood glucose, we are going to be releasing glucagon, indicating low levels of glucose in the blood, and this is going to directly act on the liver. And so this hormone will activate our protein kinase A. And so essentially we are starting gluconeogenesis as to make more glucose because we are at low blood concentrations of glucose. We have released glucagon, which will activate protein kinase A, which we've covered previously. How do we want to change our regulation of fructose 2,6-bisphosphate? So essentially, we are going to want to lower our concentrations of fructose 2,6-bisphosphate as high concentrations of F26BP will inhibit gluconeogenesis. So essentially, we are going to want to convert fructose 2,6-bisphosphate back into fructose 6-phosphate. So how do we do this? Via the bifunctional enzyme, which is the enzyme that directly acts upon fructose 2,6-bisphosphate, we know that the protein kinase A will act on this bifunctional enzyme. So we know that the protein kinase A that glucagon activated will also act on or phosphorylate PFK2 and the FBPase2. Specifically, the phosphorylation of this enzyme will activate the FBPase segment of this enzyme. And because we have activated the FBPase segment of this enzyme, we are activating the phosphatase. Therefore, we are going to remove a phosphate from our F6-bisphosphate, liberating fructose 6-phosphate, which will in turn upregulate gluconeogenesis. Specifically under these conditions, how does this all affect glycogen? So as we mentioned before, we have low blood sugar and we've released a bunch of glucagon hormone into our bloodstream. So what is this going to do to our glycogen levels? This is in turn going to lower our glycogen levels. This should make sense in that we are told that we are in a state of low blood glucose. So our body is going to want to stimulate the breakdown of glycogen in order to increase our blood glucose. So when we are trying to lower our glycogen levels or break down glycogen, we are going to be upregulating the enzyme phosphorylase A, or glycogen phosphorylase. Essentially, these are the same enzyme. We are going to be downregulating phosphorylase B and downregulating glycogen synthase. A little bit more about epinephrine as a regulator. Epinephrine has very similar actions to glucagon, as we learned in this course. Essentially, both signaling states of glucose demand, possibly low levels of glucose, low levels of energy. So like glucagon, epinephrine is going to directly activate protein kinase A. As a result, it has the action that it will lower levels of fructose 2,6 bisphosphate. And it's also important to know, Dr. Zeile points this out, where glucagon acts in comparison to epinephrine. So I just wanted to kind of write it down for you all. Glucagon will act in the liver as compared to epinephrine, which can act in muscle cells and in liver cells. All right, moving along to the pentose phosphate pathway, we learned that the pentose phosphate pathway uses the substrate glucose 6-phosphate, essentially one of the starting molecules for glycolysis, as we've learned before. So we carry out this pathway in an effort to increase cellular concentrations of NADPH in addition to 5-carbon sugars. And the reason that we do that is because this process very commonly occurs in cells where we need NADPH as a reducing agent. For example, our erythrocytes or red blood cells, or phagocytes, as I'll briefly cover later. And this process is also upregulated in growing cells that need a lot of nucleotides. As you can see, ribose 5-phosphate is one of the products of this reaction. So naturally, those 5-carbon sugars are going to be used through different types of nitrogen metabolism, which we've learned before in the course, to produce nucleotides, coenzymes, DNA, RNA. Essentially, it's important to know that growing cells will upregulate this pathway. In this process, we are carrying out 2 oxidations of glucose 6-phosphate, in addition to the liberation of 1 molecule of carbon dioxide. And like I said, this plays a large role in our red blood cells and phagocytic cells in an effort to reduce oxidative damage. So the reason that we are creating this NADPH is because it works very well as a reducing agent. So essentially, when we are in cells that contain high levels of oxygen, such as red blood cells carrying oxygen from the lungs to the tissues, we want to avoid oxidative damage of those cells as to not damage like the DNA or enzymes in our red blood cells, because that would be pretty lethal. So we produce this NADPH in order to essentially neutralize these reactive oxygen species. And we also learned that the role of NADPH in our phagocytic cells is to essentially kind of in the reverse make reactive oxygen species to kill bacteria, which is one of the functions of phagocytic cells. So that's essentially everything that we need to know about the pentose phosphate pathway for this course. Okay, last lecture before I pass this along. So I'm going to be talking now about cellular respiration, which we all know occurs in the mitochondria, which is the powerhouse of the cell. Okay, so what exactly is cellular respiration? It is the oxidation of organic molecules. So naturally, because this is an oxidation of something, this is going to be carried out in an environment that contains oxygen. We are going to be producing acetyl-CoA, and in addition to reduced electron carriers. We are then going to pass on this acetyl-CoA and the electron carriers, specifically the electron carriers, to our electron transport chain, which is where we are going to oxidize and reduce those. We are going to consume oxygen in the process. We are going to produce carbon dioxide and water, in addition to making chemical energy in the form of ATP after this entire process is done. So after traveling through the electron transport chain and interacting with ATP synthase, we're going to liberate chemical energy in the form of ATP. And I just wanted to briefly demonstrate essentially this step where we are using the pyruvate dehydrogenase complex with different interactions. It's E1, E2, and E3 subunit, which I'll explain more in the next slide. Essentially, we are using our product from glycolysis, which is pyruvate, to create this acetyl-CoA, essentially a molecule of high transfer potential. Okay, so a brief overview of our pyruvate dehydrogenase complex. In terms of its size, Dr. Zeile mentioned that it is around the size of one glycogen molecule, so it is a sizable complex. It has 3 subunits, essentially, but has many different compartments to each subunit. Its main function is to act as a one-way bridge between glycolysis and the citric acid cycle, which we'll talk about later. And this is a one-way bridge because this multi-enzyme complex does not catalyze the reverse reaction. We are going to be producing acetyl-CoA and the electron carrier NADH. And of course, this enzyme is located in the mitochondria. Okay, more about our PDH complex. Essentially, our E1 subunit is our pyruvate dehydrogenase. I would recommend that you know the names of each compartment to this enzyme. So essentially, at our E1 subunit, we are going to be carrying out the oxidative decarboxylation of pyruvate by TPP. And in addition, we are going to be reducing lipoate by using our TPP molecule. In our E2 subunit, which is also called the dihydrolipoyl transacetylase, we are going to be forming our acetyl-CoA at this subunit. And in our E3 subunit, we are going to be reducing FAD and reoxidizing the E2 subunit for the continuation of this process, essentially restoring the enzyme to its original conditions, allowing it to carry out this reaction again. In the process of reducing FAD and reoxidizing E2, we are reducing NAD plus to NADH and also liberating 1 hydrogen proton. So a little bit about the regulation of our pyruvate dehydrogenase complex. It is important to know that as we are activating PDH, we are entering a cycle in our cells that is going to create energy. It's going to eventually create ATP once we've gone through the citric acid cycle after this, and eventually moving our way through the electron transport chain. So because this is an enzyme that is going to eventually lead to the creation of more energy in the form of ATP, it makes sense that it is activated by indicators of low energy in the cell. So essentially it is activated by high concentrations of AMP, high concentrations of our CoA with the reduced thiol on the end, which is a reactant. And it is also going to make sense that this complex is is activated when it is dephosphorylated. The enzyme that does this is pyruvate dehydrogenase phosphatase. And this PDH phosphatase is activated by low ATP levels and calcium in the muscles, again, indicators of a low energy state. So overall with PDH, we are going to see enzyme activation by things that signal that we are at low energy levels. In contrast to this enzyme complex is inhibited by things that are indicative of high levels of energy. So if we have a high cellular concentration of ATP, high concentrations of acetyl-CoA, which is the product that we are creating, high levels of NADH. This enzyme is also inactivated when it is phosphorylated. It's going to be phosphorylated by PDH kinase. Kinases are enzymes that phosphorylate things. And this PDH kinase is activated by high ATP levels, as we learned in the lecture. Dr. Zeile also mentioned that PDH is inhibited by arsenic because it causes an irreversible allosteric inhibition of the E2 activity, essentially irreversibly binding to the E2 subunit of our pyruvate dehydrogenase, inhibiting the entire enzyme complex because one part of the enzyme is not working. Dr. Zeile did write out essentially the chemistry behind this on a slide. However, he told us that we don't need to memorize this. And what I would dedicate my energy towards is just knowing that arsenic is an irreversible allosteric inhibitor of the E2 activity. And here I put an example of if you all wanted to practice with the mechanism of the PDH complex. I'm not going to fill this out right now. However, if you want to test yourself, this would be a great way to do that. And I also wanted to leave you all with a few helpful videos that I recommend to my students when learning this material. Sometimes it can be helpful to just hear another person explain this content. So if you still feel like you need more supplemental material, I would encourage that you check out these videos. And with that, I am going to stop sharing my screen and we will move forward with the citric acid.
All right, so I'll share my screen for this. Okay. We are right here. Okay. So we're at the citric acid cycle. This is going to be lecture 33. And so just an overview. So like the location, obviously, as I've kind of said, is going to be important for these processes. And so it's going to occur in the mitochondrial matrix. And so other names for like the citric acid cycle are like the tricarboxylic cycle or like the Krebs cycle. So these are all like the same thing, essentially. And so you're going to be creating molecules that are going to be reduced in the electron transport chain that are essentially going to be contributing to the synthesis of ATP. So you're making like NADH, you're making 3 NADHs, you're making 1 FADH2, and you're making a GTP or ATP molecule. And so these are important because those FADH2s are going to be equivalent to 1.5 ATPs, and those NADHs are going to be equivalent to 2.5 ATPs. And so essentially what happens is you have your one molecule of glucose, and it's going to be broken down to 2 pyruvates, right? So through glycolysis. And then just as Danielle explained in the PDH complex, you're going from, you're making essentially acetyl-CoA. And so you have 2 acetyl-CoAs. And so for each of the acetyl-CoAs, they're actually going to enter the citric acid cycle. And so for each, what that means is that per glucose molecule, you're going through 2 of these cycles. And so like you can see like the products of these cycles are going to be that 1 GTP, or it can be 8 ATP, and I'll explain why. 3 NADHs, 1 FADH2, and then those like 2 CO2s from the decarboxylation. So these are the structures you guys have to be responsible for. So citric acid cycle, when you guys are studying this, is honestly not too bad compared to the other pathways because you don't have to know too many structures. And honestly, what carried me for this part of the exam was a mnemonic that I'll show you guys. And so these are kind of the structures. You guys are, I'm pretty sure, familiar with the alpha ketoglutarate and the oxaloacetate. I know it's been a little while, but I think these were the structures you guys had to know for Peerage's part of the exam for exam 2. But the two new ones are going to be that citrate and your succinate. So just study these structures, and you guys, that'll probably be like a pretty easy like point on the exam. And so in terms of the tricarboxylic cycle, so basically after you're putting like pyruvate through the PDH complex, you get acetyl-CoA, right? And so acetyl-CoA is going to come in that first step, and it's going to combine with oxaloacetate to make citrate. And so you have basically acetyl-CoA. It's going to be a 2-carbon fragment, right, of acetate bound to coenzyme A. And it's these 2 carbons essentially that can be used to extract more energy. And so if it's better to kind of think of, when you're thinking of coenzyme A, think of it as a way to handle like those 2 carbon fragments. And also just know that when you have acetyl-CoA, it's going to have something called high acetyl group transfer potential. And that's really important. I'll talk about that a little later. And so, okay, so for these, like these are obviously like all the steps for the citric acid cycle, and I'll quickly just go over the main ones that I want you guys to focus on. So if you look at it here, in terms of like, I guess the most important enzymes that I want you guys to know is it's going to be citrate synthase, and then it's going to be your isocitrate dehydrogenase, your alpha ketoglutarate dehydrogenase, and then the step right here, which is your synthetase, where you're making it. And the reason why is because these are typically the ones that are going to be the most regulated. Actually, succinyl-CoA synthetase, it won't be regulated, but the others will be. So your isocitrate dehydrogenase, your alpha ketoglutarate dehydrogenase, and your citrate synthase. And so those are all going to be regulated. And you can easily see like through here, you're making NADH here, NADH here, you're making a GTP or ATP, and then you're making your FADH2s. And so essentially you're making all these reduced electron carriers, and then we're just going to take them to the electron transport chain to convert them into energy. And so this is kind of like the overview. This is basically the entire cycle with all the enzymes. And the thing is, they won't really ask you about the chemical logic for these, so it's not really that important. You guys kind of just have to know what's going on in terms of the steps. And honestly, regulation would probably be the most important thing to study for citric acid cycle. And so what happens here is just a couple of things I want to emphasize on. So like this is a good mnemonic that I use for in terms of the intermediates. And so it's basically citrate is Krebs special substrate for making oxaloacetate. And if you think about it, citrate is that first molecule that's being made, and it's going to be a substrate, a special substrate. Essentially, it's like the first molecule you're making to make essentially your oxaloacetate at the very end. And the reason why we make this at the very end is because to use it again in the first step, like this is what completes the cycle and allows it to keep going on, is because you need oxaloacetate combined with your acetyl-CoA. And this is obviously as a result of that PDH complex. And so when these two combine, you make your citrate, and then you just go through and regenerate that to continue the cycle. And then a good one for the citric acid cycle enzymes is cheating actually is acceptable. She should forgive me. So this is just like another just weird kind of mnemonic that I found on the internet. If you guys want to use that, you guys can use that for the enzymes. But in terms of what I want to emphasize for substrate, so there's something called substrate level phosphorylation. We kind of went over this in glycolysis, but that occurs through the synthetase activity when you're making your GTP. And so what happens is actually your succinyl-CoA is a high energy, has that like high energy group transfer potential. Basically, it has the ability to transfer that phosphate to GDP to make GTP. And so you might ask, like, why do we make GTP or why do we make ATP? Like, what's the difference? Like, you can make either or. And so typically your ATP is going to be used when you have a high rate of respiration. So if you really need energy quickly, you're going to want to make ATP. And your GTP, you're going to make it if you're like a lazy cell, like say if you're a liver cell and you don't really need energy that fast, you're going to make that GTP. So that's basically the main difference of why we make either or. It's depending on the energy states. And we'll kind of see like energy states are really important in terms of regulation. I'm sorry. Okay, so in terms of hints, like so obviously, so the difference between a synthase and a synthetase, like for a synthase, you don't need a nucleotide triphosphate, meaning like you're not going to undergo hydrolysis of that nucleotide triphosphate. And so essentially no ATP or GTP is needed. But for a synthetase, you're going to need those triphosphates, and you're going to need it to be hydrolyzed in order to kind of drive the reaction. And so you have dehydrogenases again. They're going to be associated with those like electron carriers, as I kind of explained in glycolysis. And then your GTP is going to be again formed through substrate level phosphorylation. It's not going to be through oxidative phosphorylation. Typically think of oxidative phosphorylation as having to do with the electron transport chain. And I'll go over that. And so in terms of regulation, so regulation obviously is important. Typically you'll see it on your exam. And so in general, so if you think about it, through one entire cycle, so as you put per molecule of acetyl-CoA, right? So acetyl-CoA is being entered into the cycle, you're making 10 ATPs total, right? If you add it up, you're making 3 NADHs, so that's 7.5 ATPs total. And then you're making FADH2, that's equal to 1.5, so that's 9 total when you add it up. And then you have an ATP here, that's 10. So you're making roughly 10 per cycle. And so obviously if you're making energy, then those are going to act as inhibitory molecules, like any ATP, NADH, these are signals that you have a high energy state. You don't want to go through the citric acid cycle. There's no point in making more energy again, right? And so typically your activators are going to be those low energy molecules like ADP, NAD+, and then also calcium. So calcium, what's important is calcium actually, you should know which ones are going to be regulated by calcium. And a good way to just look at it is it's going to be all the dehydrogenases of the ones that I specified. So typically it's going to be your PDH, your dehydrogenase for your pyruvate dehydrogenase complex is going to be activated by your calcium. It's going to be isocitrate and also alpha ketoglutarate. So those are like the two that are, the 3 that are activated by calcium. But for the rest, they typically follow the general trend of high energy molecules inhibit. And you can easily see through, like, for example, citrate synthase is going to be inhibited by citrate. Obviously, citrate is a high energy molecule, but also it's the end product. So this is an example of just end product feedback, or like that feedback inhibition, essentially. And also succinyl-CoA. You also have to think of succinyl-CoA as a high energy molecule because it has a high group transfer potential. It can phosphorylate that GDP to GTP. So that's why we consider it a high energy molecule. And then, like, obviously NADH, these reduced carriers, ATP. And then you can see like the low ones are AMP, ADP, and stuff like that. So really know which enzymes, because they might ask you, I think I remember on my exam, they specifically asked like which enzymes specifically are regulated. So you have to know it's like essentially these 4. Okay, so amphibolic nature, they won't really ask a question about this, but I just want to quickly go over, they might ask about the main one in terms of your amphibolic, so I'll go over that. But essentially you have amphibolic and catapleroptic. And so your amphibolic associate with anabolism, your catapleroptic is going to be with catabolism. But the thing is, so the whole thing about this is literally you have a cycle, right? And these are all your like intermediates of the cycle. They're all part of the cycle. And what you can do is you can actually take one of these intermediates and kick it out of the cycle, and you can break it down. So like for example, citrate can be broken down to fatty acids, and like you don't have to know like, okay, this is broken down into this is broken down into all these. You don't have to memorize any of that. But the main one I want you guys to focus on is the anapleroptic reactions. And so that's associated with anabolism. Down, right? Because you need this in your first step, literally your first step, when you're combining with acetyl-CoA. And so if you have a low amount of oxaloacetate, it's going to be replenished through that gluconeogenesis reaction with pyruvate carboxylase, right? We're going from pyruvate to oxaloacetate through pyruvate carboxylase. That's the enzyme. And we saw that in gluconeogenesis. So that's what helps in stimulating this cycle, essentially. And so in terms of electron transport, so next I'll be covering electron transport. This is going to be lecture 34. So again, just know where like the enzymes are located. It's really important to know like in terms of like where everything's located, but also the structure of the mitochondria in general. So you have something called the inner membrane, right? You don't have to necessarily know like which part of the mitochondria is the inner, outer membrane. I mean, you can kind of tell from the name. The inner membrane is going to be more inside, the outer outside. Inner membrane space is going to be between those two, outer and inner, and then your matrix is going to be like your innermost, essentially. But like in terms of what you need to know for the for the structures of the mitochondria, is that the outer membrane is permeable to these small molecules, and meaning these small molecules can enter freely through these outer membranes. But if you have like a larger molecule that you want to enter through this like outer membrane, you need a porin. essentially. Those poron channels are what helps in guiding like the access to the mitochondria. And then for the inner membrane, so if you think about it, as you're going from like outer to inner, as you're going like deeper into the mitochondria, you have it's more selective, meaning it's harder for these ions and like molecules to get through. So the inner membrane actually is going to have a bunch of transporters, and it's going to have a bunch of transporters that allow for the transport of those small molecules, because it's not technically allowed. And you'll have like your ATP synthase and stuff. And you can see like this on slide 2 of, I'm referencing slide 2 essentially of the lecture. And then your intermembrane space is going to be that repository, that place where you're pumping your H plus ions, right, in electron transport. And your matrix is going to be where those enzymes are found. And so that's really important. And so in terms of the basics for electron transport chain, so it's important to know that it's basically just an efficient way of harvesting energy. And so what's essentially happening is you have electrons being flown like between all these carriers, right? And so as electrons flow through these carriers, you're having a bunch of free energy being harvested or being made. And so that energy is going to be used to pump protons from the matrix to the intermembrane space. One of the most important things that you guys should know about ETC is that the final acceptor is going to be O2. And so what happens is once O2 accepts those electrons, it's going to be reduced to H2O. That's really important. This is the final acceptor of the electron transport chain. And then you have your NADH and your FADH2. They're going to come from all of these pathways that we've been studying, and they're going to be reduced electron carriers, and that's what helps supply that energy. And then you'll have like iron sulfur centers. And the thing for iron sulfur centers is they basically have like a coordination complex kind of thing where they're basically accepting one electron at a time. What they do is literally, they're typically seen in all the complexes, and they literally just accept and donate electrons. They just keep the flow running for the electron transport chain. So for the first complex, there's essentially going to be 4 main like complexes that I want you guys to focus on for electron transport. And so for this one, it's going to be your NADH dehydrogenase. You don't typically have to know like what the enzyme is. Just know you have complex 1, 2, 3, and 4. And so what it does is it pumps 4 protons. So the whole pathway here is essentially you have NADH, right? This is your first step. Your NADH is going to be oxidized to NAD+. It frees up those 2 electrons. It gets picked up by something called FMN. This is a like flavin mononucleotide or something like that. It's a flavoprotein. It basically picks it up and it transfers them to the iron sulfur centers, and those pick up the electrons one at a time, and it gets transported to something called coenzyme Q. And essentially coenzyme Q picks up these electrons, and when you pick up electrons, they get reduced to QH2. So one thing I just want to know about QH2 is it's never going to leave this membrane right here, this like lipid bilayer. It's and the reason why is because it's soluble. It's lipid soluble, and so it's never going to leave this membrane. But complex 1, the main purpose is to reduce your Q to QH2, your coenzyme Q. And so you can see like ubiquinone is Q, and your ubiquinol is going to be your reduced form. And so if you think about it, coenzyme Q is in its primary state is going to be in its oxidized form, and then this is the reduced form. So that's kind of just like a picture of it. But now if you have complex 2, so if there's something that I want you guys to remember about ETC in general, besides the final electron acceptor, is literally complex 2 does not pump protons. That's probably one of the most important things. I've emphasized this in my SI sections, but I want you all to know this, that complex 2 doesn't pump protons. You'll see it on your exam. It doesn't pump protons at all. And the thing is, it's actually acting as one of the enzymes in citric acid cycle. It's going to be that succinate dehydrogenase, where you're going from your FAD to your FADH2. And so what you have here is essentially like this picture is like really like just convoluted. It's just messed up, but like you don't need to really know like the picture, but essentially what's happening is you have your succinate and you're going to fumarate, right? This is literally a citric acid cycle step, and you're going from FAD to FADH2. So what happens is these FADH2s now they're in the reduced form, they have electrons, and so they're going to transfer these electrons to coenzyme Q, and that coenzyme Q is going to be reduced. So this is just honestly another way that you reduce coenzyme Q to QH2. That's honestly like, that's the main part. And the main part is literally you're not pumping any protons. Meanwhile, in most of the other complexes, you're pumping either 4 protons or like 2 protons, which is going to be at the very end. And so that's basically that in terms of that sense. And like, if you guys reference like slide 7 on the lecture, you can see like all the complexes laid out, and you can see like the delta G values and stuff like that. And you can see easily that like it doesn't have enough. The delta G is negative 16.5. It's supposed to be like negative 30 in order to make ATP. So like, complex 2, it doesn't have enough energy in general to make ATP. So it's kind of like there on the side. And just one thing in general to focus on when you're looking at electron transport, just to notice, is that ATP synthase is technically not a part of the electron transport chain. Think of it as like a separate entity. There's a whole lecture on that too that I'll go over. So for the third complex is going to be pumping 4 protons, and it's going to use cytochrome. And so what happens here essentially is that you have cytochrome C, and it's going to be reduced. So in terms of like for what's happening here, you have your QH2, and that QH2 is essentially going to be oxidized by complex 3. And once it becomes oxidized, it's going to release those electrons, and it's going to be picked up by something called cytochrome C. And cytochrome C is going to be reduced by that QH2 essentially. And then what it does is it's going to pick up electrons one at a time because it has this heme. And the heme allows it to pick it up one at a time and not a pair. So one of the differences that you'll see between FAD and like NAD is literally FAD will accept electrons one at a time, where NAD plus will accept it as a hydride, as 2 at a time. So those are like the main differences. And like same for like cytochrome C, since it has a heme, it's going to accept it one at a time. And so you're reducing essentially cytochrome C, and you're just keep passing it on to like these different carriers. So now for the last complex 4, you're going to have you're going to deposit these electrons into complex 4, and you're pumping 4 protons into the intramembrane space again, right? For every 2 water molecules made. So this is kind of like a little confusing in terms of the mechanism that's going on, but I'll kind of briefly explain it. But what's going on here is you have electrons from cytochrome C. They're going to deposit those electrons into something called like copper A, right? This is like a binuclear copper A, and it's going to be in subunit 2. And so once it donates it to copper A, it's going to go to copper B in subunit 1. And so this is heme A, and this is heme A3. And so once like the electrons pass through here and reduce these hemes actually serve as a binding site for oxygen. Like oxygen comes and makes this like bridge essentially. And so what happens is once you make that bridge, more electrons will flow through and allow that bridge to be broken down essentially to 2 OH molecules. And then with those 2 OH molecules that get broken down, you have these like free floating H pluses here in the matrix, and they literally come in and bind to those 2 OHs that you make. And obviously with like those 2 OHs, you're making 2 waters, right? With the combination of OH and H. And so you're making 2 waters essentially. And so that's what's going on here. Subunit 3 is not like important, so just ignore that. But these are like the 2 main ones you guys have to know. And just know that complex 4 obviously is the last part. And if you look at it, obviously once oxygen binds, it's gonna bind, and then it's gonna accept those electrons, right? And so it's gonna be that final electron acceptor. And then obviously we're making water because that O2 gets reduced to the 2H2O. And so that's essentially what happens. And so essentially this is kind of like a breakdown, but for every NADH, right, that you have, 10 protons are going to be pumped into the intramembrane space. And so 4 are going to be from complex 1, none from complex 2, really important, 4 from complex 3, and then 2 are going to come from complex 4. And it's because you have 2 cytochromes, and so they need to be reduced like one per QH2 essentially. And so only 2 will be pumped into the intramembrane space, and only 1 water molecule will be made essentially per molecule of that QH2. And so what you have here is basically the movement of electrons are going to lead to something called a proton gradient, right? A proton gradient is really important because, and what that basically means is you have a high concentration of H plus in your intramembrane space and a low concentration in your matrix. And so obviously high to low, like the ATP synthase will use that difference in gradient in order to make ATP. And so in terms of like, this is kind of just like another overview of like what's going on in the overall process of basically the electron transport. But basically, and also this is just another summary. So you can see like literally NADH, this is complex 1, right? Complex 1 is going to reduce it to QH2. This is complex 2. And then you can see complex 3, where you're transferring the QH2 to cytochrome C, and then cytochrome C goes to technically oxygen to make water. And so this is complex 4. So this is just like a good kind of flow chart if you really want to simplify things. Really look at this, and helps kind of think of the bigger picture of what's really going on. And you're literally just transferring electrons at the end of the day. But for each of the complexes, I would honestly know like the different characteristics, like which one has cytochrome. Obviously, it's going to be complex 3. Things like the complex 2 doesn't pump protons and stuff like that. And also, just lastly, before I go over lecture 35, I just want to quickly say that in terms of, you might get a question on, I know there wasn't a slide on this, but if you guys reference slide 8, you guys might get a question on the inhibitors for electron transport. So I would know like what each molecule, what each inhibitor inhibits, like which complex. So like you have rotenone, which is complex 1. You're going to have antimycin A, which is complex 3, and then you're going to have cyanide, which is complex 4. And so if they ask a question like, for example, which of the following is most potent for humans, right? Like which of the following inhibitors for electron transport would be the most lethal for human beings? It would be that last one, cyanide. And if you think about it, it's because oxygen is that final electron acceptor. So it's blocking complex 4 from occurring. So you stop basically like the electron flow. So you're not making any energy. And so that's obviously going to be super lethal for humans. And so that's just like another thing to point out. But that might just be like a question you guys might see. But anyways, now in terms of lecture 35, they love asking questions about this lecture because it goes over ATP synthase. And ATP synthase is really important. And if you guys are also studying for like the MCAT and stuff like that, this is like, honestly, everything that we've covered today is going to be on there. So just some extra motivation if you guys are also studying for that. But anyways, for definition's sake, they like asking questions about what certain things are, like certain things such as like the proton motive force. And so proton motive force can be broken down into like kind of like two things. It's basically going to be the energy stored in the gradient of protons. So what that means is, if you think about it, there's going to be chemical potential energy and electrical. So if you see like the differences between, you have a bunch of H pluses here in your intermembrane space and less here. So obviously this is your P side, this is your N side. If you think about it, your P side, your positive side, more positive, and N side is going to be more negative. You can see that basically chemical potential energy is seen through the difference in chemical species, like the number of protons that you have. You have a difference, right? This is chemical potential energy. And then you can also see electrical potential energy through the separation charges. Like obviously there's so many charges here and less charges here. So you can see that there's a difference in charges essentially. And that leads to electrical potential energy. So I would know the definition of this, and then also the chemiosmotic model. That's something that's also frequently tested. I believe, yeah, this is the slide. And so chemiosmotic model, what that basically is what I've kind of been talking about for electron transport, is that as electrons flow through, right, we're actually going to be having energy released, and that energy will be used, that free energy will be used to create a proton gradient. And so, and that energy essentially is going to be used to pump those protons, which makes that gradient essentially. And so the dissipation or the breakdown of the energy in the gradient is going to be due to ATP synthase. So I know they reference ATP synthase as complex 5, but just think of it as ATP synthase. It's not technically part of electron transport. And so you have the breakdown of it, because for every, when you're making ATP in general, like H plus is going to be going from high to low concentration, since you have a gradient. And so as you're decreasing the amount of, you're increasing the amount of H pluses that go from your intermembrane space to your matrix, there's going to be less in that intermembrane space. So obviously you're dissipating you're breaking down the energy in that gradient, if that makes sense. And so for experiments, okay, so I believe okay, I can quickly just go over the experiments. This is a lot. I just don't want to go through like all this, because this is obviously just everything that's on the slide. But basically, so for experiments, you have to know experiments, unfortunately, but they're not too bad. And the thing is, for each of the experiments, they require intact mitochondria, and you require succinate. So succinate in the experiments are going to be the source of electrons. Think of them as the source of electron flow, like the source of electrons that lead to that flow. And then your ADP and PI obviously are substrates for ATP synthase. These are like our synthesis. So these are like your reactants. And then your CN, your cyanide, is going to be the inhibitor. If you guys remember, this is the inhibitor of complex 4. And then your ventricidin or oligomycin is essentially going to be your inhibitor of ATP synthase. And then DNP is going to be your uncoupler. Think of it as an uncoupler. So you have to know like what each of these functions are. And so that's important. And so with those in mind, now I just want to go over those two experiments, those like these graphs essentially, without like kind of just going over all this. But if you just look at the graph, right, in general, the whole reason why we do these experiments, if you want to reference, this is going to be slide 5 on lecture 35. And so the reason why we do this experiment is to show that you need both electron flow and you need, like without electron flow, you won't be able to make ATP. Like they go hand in hand. They're coupled reactions. And so you can literally see it here, like if you treat O2 consumed as electron flow, you can see that when you're adding the substrates for ATP synthesis, right? So you're adding ADP and PI. These are obviously needed to make ATP. And then you're adding succinate. Succinate is your source of electrons. So you have your ATP and your source of electrons. Obviously, your ATP being made is going to like jump, right? You literally have a jump in the curve. This means that you're making ATPs. However, when you add CN minus, this is your like inhibitor for complex 4 of your electron transport. This cyanide right here is literally going to stop the electron flow. And so this black line right here is going to be your electron flow. So it literally just plateaus, right? And so as this plateaus, you also see as a result, this ATP is going to stop as well. So you can see that like as your electron flow stops, your ATP synthesize is also going to stop as well. So that's why they go hand in hand essentially. And then in terms of the next experiment, you have, they switch the order, essentially. They switch the order of your succinate and your ADP PI. And so they added the electrons first and then your substrates, and then you can see ATPs being made. However, when you add an inhibitor of ATP synthase, you stop making ATP, right? Obviously, without synthase, you don't make ATP. So you're stopping it, and then you can see like literally the electron flow also stops as a result. And then until you add an uncoupler, the electron flow won't continue. So once you add that uncoupler, what it does is it continues and allows that electron to flow, like the electron flow to occur. So you have an increase in flow. However, you don't have an increase in ATP being made. And the reason why is because you still have that inhibitor. And so that you need both essentially. And that's why one affects the other. Like you can easily see that. And so I'd kind of practice going through these curves, and actually they like asking hypothetical questions like about these experiments. So for example, a question that I had on my exam, and like the only way I kind of got this was literally attending office hours. It was kind of funny because I literally saw it like right on the exam as well. But essentially when you have electron flow, right? If this electron flow continues, because when you add like DNP, like what happens? Obviously with electron flow, you have energy, right? Energy being flown, but what is that energy being converted to? So you have to know that it's being converted to heat, essentially. And that was basically like a question that I had, and that's literally just something I got from Ziley's office hours. So, but yeah, that's just like an example of a hypothetical question that you guys might get. He likes asking hypothetical questions. And so lastly, this is just a ATP synthase kind of video. If you guys want to watch just to supplement, I know like this stuff might help, because just, you know, hearing another person kind of teach might help in terms of just solidifying stuff. So definitely look at that. And yeah, that's about it from me.
Awesome. I'll take over for lectures 36 through 38. Let me share my screen real quick. Yeah, so for 36, we talked about lipid metabolism. And that's going to be kind of how or why we're using fats and how we're going to use them. So first, the reason why we're using fats is because they're very highly reduced, right? So they have more than twice the available energy of carbohydrates. And we'll look at the numbers and we'll compare them side to side. So triacylglycerol's tags, they're going to aggregate in droplets. So that's not going to raise our osmolarity of the cytosol. It's very important in terms of regulation. They're also going to be unsolvated, and they're going to weigh much less than the equivalent amount of energy in a carbohydrate. So that's really good for us so that we're not carrying a whole bunch of weight. So again, it's a very important source of energy. Our liver, our heart, our skeletal muscle, they're all going to use it. And while carbs are going to provide us with about 4 kilocalories per gram, our tags are going to provide us with 9 kilocalories per gram. So that's a lot. It's going to be a lot better for us. And tags are also going to be transported in chylomicrons. And if you guys remember, chylomicrons is basically just once you're going to emulsify through your bile salts and all that, and then you get to your tags, you're going to use your tags and combine them with apolipoproteins and cholesterol to make these chylomicrons so that you can transport these tags. So I have a question for you guys here real quick. Yeah, read through it, take your time, and then just put your answer in the chat. Let me see if I have any answers here. Okay, so no answers so far. Okay, so we got some B's in here, yeah. So that's going to be correct. So yeah, so B is going to be our correct answer. This is basically just knowing about hormone sensitive or hormone sensitive lipase, basically, and how by activating PKA, which is that whole cascade, that whole GPCR cascade, we're going to activate our hormone sensitive lipase. And the direct effect is going to be increasing our fatty acid oxidation, because that's what our hormone sensitive lipase is going to do. So again, it's going to be activated by phosphorylation. You can remember that because it's activated by PKA. And we have our free fatty acids that are flowing around. They're going to be released to the blood from our adipocytes. And glycerol is going to be released as well. It's going to be absorbed by the liver and phosphorylated. And this again works through our classic GPCR pathway that we studied for exam 2. So think about glucagon. That's going to be what's doing this for us here. For lecture 36, we talked about ketogenesis and fatty acid synthesis. So basically, these are the 3 molecules, the 3 ketone bodies that we need to know for this exam. For most of these kind of side, not main pathways, like we're not talking about glycolysis or gluconeogenesis, we don't need to know structures and things like that. But in this case, for these 3 ketone bodies, we should definitely know acetone, acetoacetate, and beta hydroxybutyrate. So ketone bodies, basically, they're going to be located in the synthesis or in the mitochondria. They're going to be synthesized in the mitochondria of the liver, and they're going to be our fuel source for tissues that are not in the liver. So when there's no glucose, we're going to use these ketone bodies. And remember that ketone bodies are going to be required by tissues and produced by the liver when you're fasting, or when you haven't eaten in a long time, when you're on a low-carb diet, or you've been working out a lot, or you also have untreated diabetes. So the way we're going to do this, the way we're going to get to these ketone bodies is through ketogenesis. So with the very first step, we have 2 acetyl-CoAs, and we want to put those together into one molecule. So we're going to use a thiolase to basically get that CoA-SH out and merge these 2 acetyl-CoA molecules together to have acetoacetyl-CoA. And once we're there, we're going to synthesize our HMG-CoA molecule, which is going to be this big bulky molecule right here. So we're going to use HMG-CoA synthase to do this, and this is by adding our 3rd acetyl-CoA in this case, and water as well, and again getting a CoA-SH out. And now we have our HMG-CoA, and that's going to be our key intermediate. It's going to be very important because our next step here is when we get that first ketone body. So by using HMG-CoA lyase to get one acetyl-CoA group out, we're going to get our first ketone body, which is going to be acetoacetate. And our acetoacetate is actually going to be very unstable. So that's why we're going to go further in this process and either do acetoacetate decarboxylase to get rid of a CO2 group and have our acetone, or we could also do alpha hydroxybutyrate dehydrogenase, beta hydroxybutyrate dehydrogenase, put an NADH in, get an NAD plus out, and now we have our beta hydroxybutyrate from acetoacetate. And basically, HMG-CoA lyase, I think, is probably the most important of these enzymes. It's going to be only present in the mitochondria of the liver, and it's going to catalyze that removal of the acetyl group or acetyl-CoA group to yield the first ketone body. So we can also go the other way, right? We can make our acetyl-CoAs back from our ketone bodies. So our ketone bodies generally are going to be transported from the liver to extrahepatic tissues, and one ketone body is going to yield 1 NADH, which converts to 2.5 ATPs. And remember, 1 FADH2 is going to convert to 1.5 ATP. And it's also going to yield 2 acetyl-CoAs, and those 2 are going to later enter the citric acid cycle or Krebs cycle, and each one of those is going to give us 10 ATPs in that process. So these are going to be metabolized in our mitochondria. Think about your muscle, your heart, renal cortex, and the brain cells. And this beta ketoacetyl-CoA transferase here is what's going to give us our acetoacetyl-CoA that we saw in the beginning here when we were first doing the first step of ketogenesis. And that's basically, we're just going backwards from hydroxybutyrate, using the same enzyme to give acetoacetate, and then doing kind of a shorter way without having to go through HMG-CoA, just using this one enzyme that's going to be only present in the extrahepatic tissues, not in the liver, because we don't want to have the same enzymes in the same place, because then we're just doing the opposite, right? We're trying to avoid this futile cycle from happening, right? Because we don't want to make ketone bodies and break them down in the same place. And then this enzyme is also going to enable the breakdown of those ketone bodies, again, to have our final acetyl-CoAs. And I know I had a question before about thiolases. So we see here that a thiolase is going to put two molecules together, two molecules of acetyl-CoA together. And in this case, our thiolase is going to break that molecule down into two acetyl-CoAs. So it kind of works both ways. It just depends on the process that you're doing. So it's kind of a heavy slide here, but ketone bodies during fasting and starvation, basically think about when you're not eating a lot of carbs, you have low blood glucose, glucagon is going to be released, our hormone-sensitive lipase are going to be activated through activating the pKa, that's going to phosphorylate them, and then our adipose tissues are going to release fatty acids to the liver. And at the same time, glucagon, because it's being released, it's going to activate our gluconeogenesis pathway, which means that oxalic is going to be used up, and that's going to slow down our citric acid cycle from happening. And in the liver, at the same time, you have these long-chain fatty acids that are going to go through beta oxidation and form our acetyl-CoA. Now, we can't really put our acetyl-CoA to the citric acid cycle because of the shortage of oxaloacetate, because we're using oxaloacetate to make glucose because we have low blood glucose levels. So what happens to all this acetyl-CoA that's in our system now? We use that to make those three ketone bodies that we just talked about. So in summary, under low blood glucose levels, glucose is going to be made using oxaloacetate. That's going to fuel tissues that require glucose and ketone bodies to fuel the rest of the tissues that can metabolize it. So just fatty acid synthesis overview real quick. That's going to be a very energy expensive process. You have to go through a lot of different cycles in order to make your fatty acid. That's because it's a very large molecule, right? You think about how many carbons you have in a fatty acid, and it's very big. Most cells can synthesize these fatty acids, and pretty much the sites that do this the most are going to be in your liver and adipose. And this is, I think, one of the special ones where we don't see that it's in the mitochondria. It's actually going to be in the cytosol. Biggest complexes is going to be our fatty acid synthase. And our final product is going to be palmitate or palmitoyl-CoA. So the formation of malonyl-CoA, that key intermediate for fatty acid synthesis, is going to happen through this enzyme ACC, or acetyl-CoA carboxylase. And this enzyme is going to be so important that we're going to have to allosterically regulate it. And basically, because it's such an important step in making fatty acids, it's going to be our rate limiting step of fatty acid synthesis, and that's why we have to regulate it very heavily. So first we have our acetyl-CoA present right here, as we can see. We're going to also have a bicarb group, and we're going to have an ATP group. Those are going to work together to become activated and form a carboxyphosphate intermediate. Once we have this carboxyphosphate intermediate, we can work through that through biotin or B7, which is going to be a very important cofactor in this process. And our transcarboxylase activity is going to transfer the activated CO2, basically, and it's going to transfer that to make our malonyl-CoA. So we're going to take it from our acetyl-CoA to our malonyl-CoA, and that's what's going to make our malonyl-CoA at the very end. So what we're doing, if you just think about transcarboxylase, you're just moving a CO2 group from acetyl-CoA to malonyl-CoA to just make this final intermediate that we're going to use later on in making fatty acids. So this huge complex I was talking about, fatty acid synthase, it's going to have 6 enzyme activities. So it's going to have its just regular synthase activity. It's going to contain a reactive thiol that's important in the transfer of acyl groups. It's also going to have transferase, dehydratase, 2 reductase activities, a thiolase, and this ACP activity as well. So we're gonna have a lot of things here. And I think it's really important to know these just so you can, when you're going through, when you're thinking about it, you can be like, oh, I know it has 2 reduction activities basically. So 2 times throughout this pathway, things are gonna get reduced, okay? Or I know it has a dehydration step, so water has to be taken out at some point. So basically going through these steps, we're going to prime it first with acetyl-CoA. So the priming that happens is going to happen right here. And then we bind with malonyl-CoA. And basically once we have that, we can start the reduction process. So the reduction process is going to take that beta keto form that we have, and that's going to transfer by putting that NADPH and getting NADP plus out. That's going to be our first reduction process. So you're just putting a hydrogen in, you're moving those electrons from NADPH to our molecule, our beta keto form molecule. And then once you have that, you can dehydrate. So take H2O, sorry guys, H2O out. And once you have that, you do your second reduction step. And I think the way I used to remember, I would just remember like the first letters of each of these steps. So P, R, D, R, T, and then the very last step, you're just basically you keep adding more carbons onto your fatty acid until you finally have your final count for your fatty acid. So basically, yeah, you have the reduction, and then you transfer the butyryl group from ACP to KS, which is the synthase activity. And I think this slide is good to look at and get the gist of it, but also the lecture slides are really good because they show you each part of the complex and how it works, and they show you a lot of the different activities that it has. So KS, you would know that's synthase activity. And then after that, yeah, our malonyl-CoA is going to be transferred to the ACP, which basically just moves around to the different places in the enzyme. So depending on the activity that you're doing, it's going to take your fatty acid, is going to move it around. And then finally, you're going to just keep adding those. So you're going to repeat it until you have your final palmitoyl-CoA molecule. So basically, from this, we gathered that we need NADPH, right? So how do we get this NADPH needed for our fatty acid synthesis? So we're going to do this in 2 ways. We can take malate and basically put NADP plus and get NADPH out through malic enzyme and have pyruvate and our CO2 molecule out. So we can do that to get NADPH and put it into a fatty acid synthase, or we can also put our glucose 6P right through the pentose phosphate pathway and get our ribulose 5-phosphate, and that's going to also get us our NADPH. It's going to get us 2 NADPHs. So lecture 38, regulation of fatty acid oxidation / synthesis. So I have a quick question for you guys. Take your time, put it in the chat. Okay, so I'm getting a couple B's here. And that's perfect. It's going to be B. So basically, for ACC, acetyl-CoA carboxylase, we know it's going to be inhibited by phosphorylation and activated by dephosphorylation. I think that's one of the most important things for this exam is knowing which enzymes are activated or deactivated by phosphorylation. And that's going to help you out a lot in terms of even when we're talking about glycogenolysis and the A forms and the B forms of the different enzymes, all that stuff. So it's very important. And then our second choice here, or number 2, says it's inhibited by glucagon and epinephrine, so that's also going to be true. And then it's going to be activated by citrate, because think about if you have, basically glucagon is just telling you that you want to make your glucose, right? You want to break down that glycogen to glucose, and then citrate is telling you, okay, well, you already have all that stuff, right? You don't need to do that. But that's going to be our 1, 2, and 3. And then 4 is just telling you that it's going to be activated by palmitoyl-CoA, which doesn't make sense because we know that palmitoyl-CoA is going to be the very last step, the end product of using ACC and getting that malonyl-CoA. So a regulation of fatty acid synthesis, we know that ACC is going to activate fatty acid synthesis because it gives us that malonyl-CoA molecule. And it's going to be inhibited by phosphorylation, like we said. So pKa basically is going to inhibit it, and the way you get to that is by using glucagon to get to that. Also, it's going to be inactive due to low energy status. So basically, when you don't have enough glucose, you release glucagon, and that means your pKa is going to be activated. It's going to phosphorylate your ACC. So that's why if you get a question that tells you you have low energy, are you going to be making fatty acids? No, you're not going to be making fatty acids. Also, palmitoyl-CoA, like we said, it's its product, so it's going to inhibit it. And activating it, we could just dephosphorylate it and also use citrate to activate it. So for regulating our TAG synthesis in the liver via hormonal control, so let's say you have high blood glucose, right? High blood glucose is going to tell you to release the hormone insulin. Insulin is going to work to promote the pathway of glycolysis, break down that glucose because you have a lot of it. And once you get pyruvate from doing glycolysis, you can take that through PDH complex like we know, get that acetyl-CoA, and then make those ketone bodies and fatty acids. Now, on the opposite side, where you have low blood glucose, you're going to, this should say glucagon, right? So you're going to release the hormone glucagon. That's going to activate your pKa. That's going to make your ACC inactivated through phosphorylation, which means you won't be making any fatty acids. And these are different ways this can happen. So you're intaking dietary carbs, that's a lot of glucose right there, or you're intaking dietary proteins, it's a lot of amino acids right there. So you go through here, you go through glycolysis, it's going to be upregulated because of this high glucose intake and this insulin release as well. So that's going to be upregulated. You're going to have acetyl-CoA and basically go make your ketone bodies or go make your fatty acids, depending on the phosphorylation of your ACC enzyme that's going to make your intermediate of malonyl-CoA, and your fatty acids are then going to bind with a glycerol molecule to make your tags, and from there you can store them. You could emulsify them and make your chylomicrons and distribute them throughout your body. Just depends on the scenario that you're in. I think those are really good to know for the exam in terms of scenarios. I think that's it. I think I'll pass it on to Matthew now.
Can everybody see the screen? We're good?
Yes.
Yeah, cool. So we're going to be going over the last two lectures, lectures 39 and 40. So beginning with cholesterol synthesis. So the location of cholesterol synthesis is the cytosol, right? As Kishan talked right in the beginning, that's very important for you guys to know exactly where these reactions are taking place, okay? So for cholesterol, is obviously going to happen in the cytosol. I apologize. And one important thing to note is that for the beginning of the cholesterol synthesis, it looks very similar to how ketogenesis works up until HMG-CoA. Okay, but just make that distinction that ketogenesis is going to be occurring in the mitochondria, while cholesterol is going to be occurring in the cytosol. So there's 4 main stages that Zali talked about, where we're not going to go over all of them. We're just going to be giving a big overview of what cholesterol synthesis is. So we're going to be starting off with acetate, and then we're going to move on to mevalonate, then activated isoprene, squalene, and later cholesterol, okay? So obviously acetate is formed from our acetyl-CoA, okay? And then it will be used to form this mevalonate. the mevalonate will become this activated isoprene, okay? It's gonna be, you're gonna start seeing that when we're moving from activated isoprene to squalene, we're gonna start making these cyclic natures right here, okay? Then squalene going to cholesterol is gonna have a series of multi-step reactions that you don't need to know. But an important reaction that you should know about the conversion of squalene to cholesterol is the addition of oxygen into squalene that drives the cyclization reactions that eventually form cholesterol. That's very, very important, okay? But for the main purpose for this exam, just be aware of these intermediates of the cholesterol synthesis. And for example, the main rate limiting step, which is going to be our HMG-CoA reductase, right? So when we're going to be looking at regulation of cholesterol, it's a lot, right? But as you're going to see, as I just talked about, HMG-CoA reductase is the most important enzymatic step on the cholesterol synthesis pathway, okay? And this is all going to, HMG-CoA reductase is how we convert acetyl-CoA into our mevalonate intermediate, okay? So as we're going to see here, HMG-CoA reductase is inhibited by phosphorylation, okay? So there's two key kinases that affect HMG-CoA reductase. We have our AMP kinase, which in the graph here, it's designated as AMPK. And we have our protein kinase A, which is activated by glucagon, right? As we know that glucagon activates through our GPCR, which eventually activates a PKA, which would eventually phosphorylate HMG-CoA reductase. And that also makes sense, right? Why these two species are going to be inhibiting HMG-CoA reductase. AMP kinase is only activated in low ATP situations, right? So why would we try to make cholesterol if we don't have enough ATP? It doesn't make sense, right? On the other hand, if glucagon is indicating that we have a low blood sugar, so why would we start making cholesterol if we need more blood sugar to be released into our blood vessels? So that's why glucagon would be inhibiting HMG-CoA reductase. In terms of activating, we have one major activator that actively activates HMG-CoA reductase, and that's insulin, right? Insulin indicates to our body that we have a high amount of blood glucose. So we need to find a way to remove as much of these glucose or these energy molecules as possible from our blood. So a good way that we can do that is also by forming cholesterol, right? Because it reduces acetyl-CoA from our bodies to allow more glucose to be converted to acetyl-CoA. So insulin would actually activate a phosphatase, which would dephosphorylate HMG-CoA reductase, activating HMG-CoA reductase to eventually allow the cholesterol synthesis to occur rapidly, okay? So in terms of other ways that we are able to inhibit HMG-CoA reductase, we also have the oxysterol. So oxysterol is just the oxidation of cholesterol molecules in our body. So if you have a high amount of cholesterol molecules circulating in our body, there's a very, very high chance that these cholesterol molecules will be oxidized to form these oxysterols. So this is one way that our body indicates to our HMG-CoA reductase that we have way too much cholesterol and we can stop the synthesis pathway. Another way that we're able to activate HMG-CoA reductase, although it's kind of indirectly, is through the ACAT, or the acyl-CoA cholesterol, or acetyl-CoA cholesterol, or acyltransferase, which will catalyze our cholesterol into cholesterol esters. As we start forming cholesterol esters, we start diminishing the concentration of cholesterol intracellularly, allowing cholesterol synthesis to occur more rapidly because we don't have as much products, right? For example, as we remember from exam 1, that Le Chatelier's principles, right? If we have way less products than our substrates, our reaction is going to move towards our products. So if you find a way to remove as much cholesterol as possible, we're going to be able to keep making that cholesterol synthesis work, right? And another big example as well is how oxysterol can also inhibit our receptor-mediated endocytosis. As we're going to see, a lot of our lipoproteins work through this receptor-mediated endocytosis. So if our cells have a lot of cholesterol, we need to find a way to prevent other lipoproteins from approaching us and giving their cholesterols to us. So now moving on to lecture 40, our plasma lipoproteins. So what is the purpose of a lipoprotein, right? So they're basically a ball of fat surrounded by protein. That's how I like to describe it sometimes, because this is going to transport cholesterol esters, triacylglycerols in the aqueous environment for the blood circulation, okay? So these are made up of apolipoproteins, which are the proteins that surround the lipoproteins, okay? But the big plasma lipoproteins that you have to know are our chylomicrons, our very low density lipoproteins, our low density lipoproteins, and our high density lipoproteins. So for our low density lipoproteins, these are large macromolecular complexes. As our very low density lipoprotein starts losing our triacylglycerols in the circulation, it starts forming into these low density lipoproteins, okay? And low density lipoproteins have kind of a bad reputation. They're called usually the bad cholesterol. And that's because low density lipoproteins have a high amount of cholesterol in them, okay? And they're usually the ones that will start donating their cholesterols to different tissues such as our adrenal tissue, our muscle tissues. So they carry a lot of cholesterol. So if you have a high amount of low density lipoproteins circulating in our blood, it could mean that it's indicating that we could have cardiovascular disease or other big issues such as stroke, okay? So a big important differentiation from low density lipoproteins from everything else is that their key protein here is our ApoB 100, okay? So be aware that it's ApoB 100 because that could be an easy question that you could miss just because you confuse the names of the apolipoproteins, okay? So an important thing as well is our low density lipoproteins are surrounded by a single layer of phospholipids, not a bilayer. And the lipid composition of our low density lipoproteins constitutes phospholipids, cholesterols, cholesterol esters, and triacylglycerols. So here's just a graph explaining all the different plasma lipoproteins and where they're formed, which is also very, very important. Where they're formed, their functions, for example, which pathways they go towards, right? Our exogenous, our endogenous, or our reverse transport, and what they usually mostly contain, right? So just a reminder that we talked about chylomicrons, I think in the second exam, if I'm not mistaken. Remember that chylomicrons are formed in our intestinal cells, and they're the ones that grab as much of those triacylglycerols as possible from our intestines, okay? So chylomicrons are kind of separate from all the very low density lipoproteins, for example, from all the other density lipoproteins, but they're also very, very essential, right? Because they're the ones that are going to be collecting our triacylglycerols from our intestines, okay? But everything else here is super essential, right? So our very low density lipoproteins contains a lot of triacylglycerols and cholesterol. Our low density lipoproteins are going to be the ones that are mostly going to be transporting our cholesterol to the rest of our bodies. And our high density lipoproteins are almost like our recyclers, right? They're going to be collecting free floating cholesterols all around our body and bringing it back to the liver to be reformed into something else. So now we're going to be looking at different pathways. So for the exogenous pathways are the ones that involve our chylomicrons, right? They're going to be in the intestines. And they're going to be the ones that are going to be picking up as much triacylglycerols as possible and depositing them in adipose tissues for the most part. Okay, so as you see here, they're the ones that are going to be giving out these free fatty acids to our adipose tissue. And then our chylomicron remnant, when this remnant is when our chylomicron doesn't have anything inside them at all, their function is done. They're just going to move back into the liver, where they're either going to be converted back to cholesterol, or their free fatty acid, there are still some are still left over, are going to be given back to our livers. For endogenous pathway, this is the one that's a little bit more complicated. Our very low density lipoproteins are going to be filled with free fatty acid and cholesterol. They're going to move about our bodies, right? They're going to give the free fatty acids to our adipose tissue. And then we're going to be forming into our low density lipoproteins where they have a lot of cholesterol, right? And they're going to be giving out these cholesterols to our peripheral tissue, okay? Through these LDL receptors that we're going to talk about right towards the end. And then for our reverse cholesterol transport, this involves our high density lipoprotein, right? So as we see here, an empty high density lipoprotein will move to a peripheral tissue that might have a lot of low density lipoprotein, so that has a lot of cholesterol, and these high density lipoproteins are going to able to move these low density lipoproteins or the cholesterol that's inside them back into the liver, right? So it's just a recycler. So we want a lot of high density lipoproteins in our body when compared to low density, right? Because high density just cleans up the extra cholesterol in our tissue. So for our receptor mediated low density lipoproteins, so this is actually a really cool receptor and pathway. As you see here, our low density lipoprotein, as we know, has the ApoB 100 apolipoprotein attached to around the big region of the lipoprotein, right? and it's going to attach itself to the receptor of a cell. Okay, and this is going to initiate an endocytosis reaction, right, or endocytosis pathway. This is the LDL is going to be formed into this endosome. Later, the receptors that are still attached to the LDL are going to be removed and brought back into the cell membrane. Later, that endosome is going to join up with lysosome, where it's going to start breaking apart that LDL into its basic components, right? So it's going to give us mostly cholesterol, but it can also give us some fatty acids and amino acids as well. Okay. Oh yeah, so this was the last part of the lectures, but these are just a little reviews that are still at the end. So this is just the difference between our mitochondria and our cytosol. So this is where some of the reactions occur, right? So our PDH, citric acid cyclone ketogenesis occur in the mitochondria, while in the cytosol, we have our cholesterol synthesis and fatty acid synthesis. And here all the structures, like Xali doesn't really care that much about structures as much as Peerage, but he still wants you to know how much, 7 structures. And for the most part, you should know some of these structures already from exam 2. So it shouldn't be that bad, but I think, yeah, that is the last slide. So we're going to stick around for a little bit, answer any questions you guys have. But if not, we've been recording this review, and then we're going to be posting it as soon as we can to our SI Slack, and then your SIs can give you guys the information on the review. But if anything, please start studying if you haven't, and good luck on your exam. I know you guys are going to do great, and thank you so much for sticking with us on a Monday night. I think I can stop the recording, right?
Yeah.